Alright, so this video is going to be about DNA going to RNA that goes on to be proteins. Remember, this is the same lecture that we did in class, but if you had a little trouble, we can definitely use this as a good review process. So again, we're looking at DNA to RNA down to proteins. So amino acids, this should be a review from first week or so of class, but we learned about amino acids and that they are the building block of proteins, so you can almost think of them as like Legos that would bind onto each other to build up proteins. And whatever Legos, whatever different amino acid is in each Lego, that's what is going to give the correct order of a protein. Amino acids have three distinct parts. So the amine group is signified by this nitrogen here. The carboxyl group is signified by the carbon and the two oxygens here, and then you have your R group, which, remember, this is what makes each amino acid different. So it makes the amino acids different, whatever is bound to this R group. So there's not actually an R there. It could be, just another carbon, or it could be a string of carbons. A bunch of different things could go there. So remember, since DNA is completely locked up in the nucleus and it can never leave, DNA will never leave the nucleus. Remember, the nucleus is pretty much just there for the protection of DNA. And DNA, if we remember, is what codes for everything in life. And without DNA, we wouldn't have the code for what we become. But since DNA is locked in the nucleus, we've got to have some way for it to get its message out into the cytoplasm. And that's where this awesome RNA molecule comes into play. Now, RNA and DNA, of course, sound very similar. That's because they are similar. There are three main differences. RNA is still made up of nucleotides, but the fact that it is made of ribose sugar instead of deoxyribose is difference number one from DNA. RNA still has a phosphate group, just like DNA does, and RNA still has 4 nitrogenous bases, cytosine, guanine, adenine, but remember that this uracil is what is different from DNA. So uracil is replaced, it replaces the thymine in DNA. So DNA has a thymine, and remember that's signified by a T. So thymine would be in DNA. RNA has uracil. Now RNA is also a single strand of nucleotides, unlike double-stranded DNA. So that's difference number 3, and the uracil up here being difference number 2. So we can see this is like a half of a spiral staircase here, and we've got our C's, our G's, our A's, and then our difference in our U. There are 3 types of RNA. Messenger RNA is the most common one that you'll hear about. So a messenger RNA has a linear shape, and it carries the code for the protein synthesis into the cytoplasm. And this is actually a exact copy. Sorry, it's a complementary copy. So I apologize. It's a complementary copy of DNA. And then you've also got your ribosomal RNA and your tRNA. So ribosomal RNA, we're going to learn, is a protein building machine, and then tRNA is the transfer RNA. It literally looks like a T, as you can see here, and it is what carries the amino acids. Like, maybe you have it attached up here and it's got, a different shape to it, but it carries the amino acid to the ribosome. Now, proteins, this should be a bit of a review from the first couple of weeks of school as well, but remember, RNA turns the genetic information from DNA into our proteins. And proteins, they're going to do so many different things. They help with metabolism, cell shape, signaling, responses, cell cycle, and they form the building blocks of life. Now remember, we talked about how there are thousands thousands of amino acids that make up one protein. That's very important to remember because all of our models that we do are only going to show maybe like 10 or 15 amino acids to make up a protein, but it really takes thousands. Here's our overview of protein synthesis. We start off with our DNA here in the nucleus, and our mRNA is copied in the nucleus, and it is sent out from the nucleus into the cytoplasm, where it then attaches onto this ribosome, and this ribosome is going to read each individual codon, which, remember, a codon is 3 base pairs, and that tRNA is going to come on with the anticodon, bind into here, and that tRNA has carried that amino acid, and the amino acids just go on to make this polypeptide chain. There are 8 steps to protein synthesis. The first one is the copying of mRNA from DNA. You can see that process here where we have our strand of DNA going through this RNA polymerase, and that's what this enzyme here is. The RNA polymerase just reads down the DNA and it kind of shoots this ribbon of single-stranded RNA through there. The ribbon is going to slip out of the nucleus and attach to a ribosome, and remember that each set of 3 nucleotides of mRNA are called codons and code for a specific amino acid. So if we have, and I do not like this picture here, I should take that out, but each part of the mRNA, here's 3 base pairs, 3 base pairs, and I'm spreading them out. They're not actually spread out like this, but I'm doing it just so you can visually see. Maybe this base pair, this codon, says AUG. And by that chart that we've learned about, you'll see that AUG codes for methionine. And then maybe we have a CUA here. I can't remember what this one codes for, but it's another amino acid, and so forth. Each one is going to make a new amino acid. The second part is called translation. This is where the mRNA is actually translated into a protein. So the tRNA is going to be floating around. This tRNA is going to be just floating around in the cytoplasm. And it's going to be carrying this amino acid up here with the anticodon. So we learned about how, if the codon on mRNA was AUG, then the codon for tRNA would be UAC, the anticodon. tRNA is going to bring the amino acid from the cytoplasm by matching its anticodon, this guy here, with its codon and the mRNA, these here. The ribosome is going to connect all additional amino acids to the first amino acid, so you can see here that that's where they start to make this polypeptide chain. Then the additional amino acids are delivered by the mRNA until a stop codon is reached. Then the amino acid goes on to be folded as a protein, and then it's going to go and make up different parts of our body, whether it be a cell, or go and help carry out an activity through an enzyme, something along those lines. I sure hope that this helped you, and please use this as a great resource for review.